stabilizers. That is what we spoke about. Today we are going to deal with about the biotransformation and bioconversion. Most of your articles also, like we see that biotransformation and bioconversion, they it is one and the same word, like that it is being used, but actually that is not the case. Biotransformation and bioconversion are both different terminology, but in most of the articles we see this, it is written as one and the same. So this biotransformation is a process, okay, where different substrates are used for the cell growth and product formation. So whenever the synthesis of desired product often only compresses one or two steps, that is known as your biotransformation. Whenever you do biotransformation, as I told you, it involves only one or two steps. Theoretically, when you calculate, okay, you get a very high yields. And furthermore, the design and synthesis of heterologous production strain in many cases is less elaborate, as often only one recombinant gene has to be expressed. So we will discuss and describe two examples of what is biotransformation and what is bioconversion is. So, as I said earlier, biotransformation is a process by which an organism or its enzyme, this organism or this organism's enzyme, bring about chemical changes on the compounds that are not part of their metabolism, and they result in the formation of novel or useful products that are often difficult or impossible to obtain by conventional chemical means. This is the meaning of biotransformation. So you are using in biotransformation, you're using an organism, microbial organisms or its enzyme. Using that, what you're doing, you're bringing about the chemical changes on components. What are these components? This components is in no way related to that organism or the enzymes of the byproducts of the enzyme. No. Okay, they bring about chemical changes on compounds that are not part of their metabolism, and they result in the formation of novel or useful products. So this organism or this enzyme is bringing a change in this compound in such a way that it produces a novel product. And this product is very difficult or almost impossible to obtain by the conventional chemical means. By conventional chemical means, by chemical treatment, you cannot bring about that new product. Whereas your microorganisms and its enzyme can bring about a change in that product such that that product will be a novel one. Okay, and it should, novel means obviously it will have a useful product. So this is known as biotransformation. So bio means it is the organism or the enzyme of that organism. What it is doing, it is bringing about transformation of what? A compound. And that compound, it is not related to this organism, but still it acts upon that compound and it converts this compound into a novel or useful product. And this conversion is not possible by conventional or traditional chemical means. That is the meaning of biotransformation. The total chemical transformation of one steroid to another not only requires many stages but an expensive process, and although provide only low yield. So when you, one example what we have told is the steroids. The chemical transformation of one steroid into another steroid, it requires many stages. Okay, So this steroid, I want to convert this into another steroid. What it requires many stages, one, two, three, four, like that, many stages. We don't know. But not only this, so many stages if it involves, it becomes an expensive process. So many steps is involved. It is expensive. And above all, you get only low yield. How? By chemical transformation. Then I don't want this, right? I can go for biotransformation. That is what we explained. Biotransformation entails the conversion of the preformed substrate into a product of choice, usually only involving one or few enzymatic steps. That is what in the starting step explanation I told you. It requires only, your biotransformation requires only one or two steps. Okay, when it requires only one or two steps, when you do theoretical calculation, okay, you get a very high theoretical yields. And also what you can do is, nowadays by using genetic engineering, you can alter this organism. So you can produce an heterologous production of strain. So what will happen? It can increase more the yield of your product, and it can bring about a better transformation of your product. So that is the meaning of biotransformation. So transformation reaction in large scale equipment are carried under sterile condition in aerated and stirred fermenter. That is the or stir tank fermenter, and it should be aerated. Of course, all will not be repeating this all once again. You have to please go back to my previous videos to understand all this. Okay, how we bring about the sterilization, what is aeration, what is stir fermenters. Okay, the conversion process being monitored chromatographically or spectroscopically. So you are bringing the transformation of the product into a novel product by a microorganism, or the or the organism's enzyme. How? In a fermenter. So you have to monitor whether the things are going properly. How you do it? By using chromatographic technique or the um, spectroscopic technique. What is chromatography? You all know that it is a separation of mixture of compounds. If there is a mixture of compounds, I can separate it by chromatography method by using the various solvents. Spectroscopically means what is the quantitative mass I am getting. That is what you do it in spectroscopy. Okay. So we monitor this by using chromatographically, that is what type of mixture is needed to be separated, and we'll quantify it, our product of interest, by spectroscopically. Once this has been done, what we do is the process is terminated when maximum titer is reached. That is, the maximum amount is reached, then we stop the fermenter. Maintenance of sterile environment is necessary because contamination can lead to the formation of faulty conversion products and also engage in total breakdown. So from the beginning, we have always told that sterilization is one of the most important part and parcel of the fermentation. If we don't look into the sterilization and we look into all other parameters, optimization, standardization, and we forget about the sterilization, look, whatever you did is waste, isn't it? So you have to maintain a sterile environment, otherwise it can lead to the contamination of your product. So contamination And also what faulty conversion, what you, if you want A component, you will be getting Z component, which is not your compound of interest at all. And it may result in total breakdown. So biotransformation involves either the whole cell or with isolated enzyme. That is the first step we spoke what is biotransformation is. So let us take few examples. That is, I will be taking two examples which can bring about biotransformation. So transformation of steroids and sterols. So steroids constitute the natural product class of compound that is widely distributed throughout the nature. It is present in the bile salt, adrenal cortical, sex hormone, insect molting, hormones, sapogenesis, alkaloids, and some antibiotics. So your steroids and are present in all of this, whether it is a human or insect or animals, you can see the presence of the steroid. So if you go into look at the history of the steroids, in 1937, the first microbial transformation of steroid was carried out. So biotransformation, I told you, bio means some biological. Here what that some biological is nothing but a microorganism is bringing about the transformation of the steroid into a useful product, a novel product. And that was started in 1937. And so testosterone, okay, which is an hormone, a male sex hormone, was produced from dehydroepiandrosterone by using Corynebacterium species. So testosterone is what? It is a steroid. Okay? It is one of the form of the steroid which was produced from what? Dehydroepiandrosterone. So this dehydroepiandrosterone was there, and this corynebacterium, okay, when it acted upon this, it produced this testosterone, which is an important steroid hormone. Subsequently, cholesterol, you have all studied about the cholesterol metabolism, and we told that the cholesterol is produced de novo and you need not take the cholesterol in your food. It is not essential because it is produced in your body. And we told that the important function of the cholesterol is what? One is the vitamin D, and then it produces the steroids. Okay. And this also was produced from 4-dehydro ethylcholinic and 7-hydroxycholesterol using Nocardia species. So in both you see production of testosterone, production of cholesterol is done by what? By bacterium. One was Corynebacterium and another one was a Nocardia species. What was the starting material? It was a different material. Here you can see androsteroids for testosterone, and here you can see this 4-dehydroethylcholinic and 7-hydroxycholesterol using Nocardia species. So this Nocardia species acted upon this, that is my organism, bioorganism acted upon this substrate and converted it to a novel product called as cholesterol. That was the what the first slide meaning was. Okay. Another example, let us take the production of ascorbic acid, which is vitamin C. It is usually produced by this Rensting-Gessner synthesis, named after the scientist who synthesized this L-ascorbic acid. The process of microbial conversion consists of several steps, and this L-ascorbic acid is used in vitamin preparation or as an antioxidant in the food manufacture. The oxidation stage from D-sorbitol to L-sorbose is carried out by Acetobacter in submerged process, and 30 to 35 degrees centigrade with vigorous stirring and aeration. That is, we are going to use the submerged method, wherein your organism will be submerged along with your product, with your substrate, okay? And the temperature is the 30 to 35 degrees centigrade, and there is a vigorous stirring and aeration. This process is carried out continuously in two stages. The first step involves oxidization of the glucose by Erwinia species. So, the first step what it involves is your oxidization. Okay, it involves oxidization of glucose by the Erwinia species to 2,5-diketo D-gluconic acid. How this is produced? Your glucose is getting oxidized to 2-gluconic acid and 2-ketogluconic acid by this organic Erwinia species, and finally what you get is 2,5-diketo D-gluconic acid. So these are the intermediate step. In the second step, it involves this 2,5-diketo gluconic acid to 2-keto 1-gluconic acid, which is catalyzed by a Corynebacterium species. Okay, so this is another example wherein we are using what microorganisms. We have used for the production of vitamin C, we have used two-step reaction. One is the Erwinia and the other is the Corynebacterium. This Erwinia, what it does is, in the first step you see that oxidization stage from D-sorbitol to L-sorbose is carried out by the acetobacteria in a submerged fermentation process at 30 to 35 degree with vigorous stirring and aeration. So next what happens, this Erwinia species, it oxidizes glucose via these two components, intermediates, to form your final compound 2,5-DKG. In the second step, your 2,5-DKG is converted to keto 1-gluconic acid, which is catalyzed by the Corynebacteria species. Also, I have given another example of prostaglandins. These are, your prostaglandins are unsaturated fatty acid. Okay, they consist of 20 carbon fatty acid hormones. This contains various medical significance, such as your prostaglandin 2 can be used in contraceptives, and PEG2 is used for alleviation of pain during childbirth, and PEG1 is used for the treatment of congenital heart failure and also for the treatment of digestive diseases. These prostaglandins can be produced from unsaturated fatty acid by microbial transformation with pathogenic fungi such as Cryptococcus neuroformans. So these prostaglandins, which have medical application, can be produced by unsaturated fatty acids, which can bring about microbial transformation into an useful component or novel component prostaglandin, which is having various functions, okay, the medical applications. Now coming to the term bioconversion. So bioconversion is defined as a synthesis of biochemical from basic nutrients by a microorganism. Okay, it is defined as a synthesis of biochemical from the basic nutrients of the microorganism. The decided product is in this case is produced by a cellular metabolism encompassing multitude of enzymes. So your bioconversion, who is bringing about the conversion? Bio means again it is my microorganism. Okay. And here how the conversion is brought about. The synthesis of biochemical. So this microorganism is synthesizing biochemicals from what? From the nutrients by the microorganisms. So the microorganisms convert these nutrients into a product that is known as the bioconversion. So the decided product in this case is produced via the cellular metabolism encompassing of multitude of enzymes. Bioconversion differs greatly from biotransformation and also from conventional chemical synthesis. We also, in the starting itself, I told you that biotransformation is better when you compare it with conventional chemical methods where you saw the yield was so low. Okay, and this bioconversion is different from this term biotransformation. Here, the microorganism and its enzyme in one or two steps it brings about the transformation of all a compound which is already present into a novel compound. Here, what is happening in bioconversion? It is a synthesis of a biochemical from this biochemical from the basic nutrients by microorganism. So the decided product is in this case produced by the cellular metabolism. Where does the cellular metabolism occurring? The cellular metabolism is occurring in the microorganism. See, even in our body, the cellular metabolism is occurring. Okay, what is bringing about the cellular metabolism? It is the enzymes. Similarly, in microorganism also you have the cellular metabolism, which is bringing, which is brought, which is catalyzed by the enzyme. And what do you mean by the cellular metabolism? This is what when I eat something, if I'm consuming that food should be broken accordingly so that I get the carbohydrates, fats, whatever I need. Similarly, the same thing, no, the microorganisms require nutrients. When they consume the nutrients, it has to be broken according to their composition. And that is why the cellular metabolism is occurring even in microorganisms. They are living fellows, creatures. Okay, so the synthesis of biochemicals. Since the same substrate is used both for catalyst and product formation, the yield is often relatively low. In bioconversion, what happens is this one particular substrate is used for both the product formation and even for the catalysis. That is why the yield is low. The use of living organism as catalyst also means there may be a greater number of side reaction and purification of the product can be challenging. So what is happening here is your substrate of the organism, it only acts as an enzyme, like an uh, enhancer, okay, pushing forward the reaction. It acts as a biocatalyst and product formation. Therefore, it is often the yield is low here. The use of living organism as catalyst means there may be greater number of side reaction. So if my reaction has to go like this, maybe I'll be having a side reaction. And therefore, the purification of the final product is very challenging in this method. But in return, the substrate is often cheaper and more readily available. From an environmental viewpoint, the greatest advantage of bioconversion is that it can rely solely on renewable substrate. Okay, so renewable substrate means that is they'll be always present in the nature. They're just the opposite of non-renewable. Okay, like petrol is a non-renewable, whereas your sunlight is a renewable. Okay, so the bioconversion, because always the sunlight is will be there, the energy whatever you're getting from the sun. Okay, the bioconversion slowly uh, the bioconversion is that it can rely solely on the renewable substrate, meaning that carbon dioxide emission from the process are captured by the plant used as feedstock in closed carbon cycles. So let us take few examples of this also and study. So we'll take the example of rhamnolipids. These rhamnolipids are amphiphilic biological surfactants composed of hydrophobic and hydrophilic moiety. They have both hydrophobic and hydrophilic natures, and hence they are called as amphiphilic surfactants. These rhamnolipids are glycolipids as their hydrophilic part is a hexosaminose. They accumulate at phase interface and therefore can be utilized as a green washing detergent. And rhamnolipids overcome two disadvantages of chemically synthesized detergent. As biosurfactants, they are produced by bacteria. They can easily be synthesized based on the renewable resources, diminishing the dependency on the fossil resources. So that is why it is called as a green washing detergent, because we are using the renewable energy. So we are not depending on the non-renewable energy. We are not taking something away from the nature, which is always remains in the nature. Only by that we are using, and that is why it is called as a green washing detergents, and it is used as a biosurfactant. And they are also degradable by bacteria and thus have potentially a lower impact on the environment. Whatever the product is being formed, it can be degradable by the bacteria, and hence it is not having any ill effect on the nature. In nature, rhamnolipids are excreted by the bacteria to increase the availability of hydrophobic carbon sources. For example, they contribute to the formation of suspension of oil-water mixture, enabling the organism to grow in crude oil reservoirs or oil tanks. They also play a role in biofilm formation and the virulency factor of Pseudomonas aeruginosa. Furthermore, rhamnolipids affect the membranes of gram-positive bacteria, thereby causing an advantage over nutrition competitors. So these are all example of your rhamnolipids which are produced by the renewable energy resources, wherein you can produce the green detergent, and whatever you have produced, the end product, okay, that doesn't add to the wastage, because they can be degraded by the organisms. So hopefully this is clear. So your green detergents are made by natural materials, that is designed even from plants also, just not by the microorganisms, even by the plants they are being produced. So this is one of the example. Another example what we are going to study is the acrylamide. So acrylamide is very important commodity for chemical which is mainly applied as monomer for industrial synthetic fiber production. It uses range of coagulators over polymers for vapor treatment to petroleum recovering agents. Acrylamide used to be produced by chemical synthesis when in 1985 Pseudomonas chloranthus B23 was found to synthesize a nitrile hydratase which could hydrate acrylonitrile and form acrylamide. So what it tells us that initially this acrylamide was produced by the chemical synthesis process, but later on it was known that this organism, the Pseudomonas chloranthus B23, was able to synthesize a nitrile hydratase which could hydrate acrylonitrile and form acrylamide. Okay, so this nitrile dihydratase is the your enzyme produced by this organism. So this enzyme is catalyzing the reaction. Which compound? This compound, hydrate acrylonitrile. So your Pseudomonas produces the enzyme nitrile hydratase. Okay, this is your enzyme and this is your substrate, and what it is forming? It is forming acrylamide. Now who brought about this conversion? It is your Pseudomonas, which is an organism. So as often enzymatic process had advantages of high selectivity over chemical process. Also the titer of acrylamide in the media was higher. So what they mean to tell is that when you compare with the chemical method, okay, the enzymatic method has more advantages. Enzymatic method means when where you're getting your enzyme, you got it from the microorganisms. And here, what was a microorganism? Pseudomonas chloranthus B23. This is a strain. And what did it produce in within its body? During biochemical synthesis in its body, it produced an enzyme. The net name of that enzyme is nitrile hydratase. If there is an enzyme, it has to act on the substrate, right? It acted on this substrate, hydrate acrylonitrile. And what did it form? It formed the product acrylamide, which was earlier formed by the chemical. Not only that, it also produces an higher titer of acrylamide, that is approximately 50%. In chemical method, you get only 30%, but here you're getting 50%. Another benefit is that the reaction could take place at lower temperature and pressure, yielding further saving the cost. This is another important using the microorganism. The energy consumption and carbon dioxide evolution decreased about 30%. So the energy consumption, the usage of energy and production of carbon dioxide decreased by 30% by using this method. The industrial production of acrylamide with recoverable immobilized whole cell catalyst. Okay, now what they have done is instead of using the microorganism, they have made it an immobilized, that enzyme, whatever, or the microorganisms, it can be immobilized. Suppose say if my microorganism or the enzyme is immobilized, that is it cannot move. So my substrate is that my substrate can enter into this and then it can form the product. So your enzyme will be here only, it can be recovered. Okay, again it can be reused. It's not necessary that along with your product even the enzyme will come. No, that is what is known as immobilized enzyme. The first step is the production of the biocatalyst, which comprises of cultivation and immobilization of the bacteria. So that Pseudomonas is the bacteria. So what you have to do is the production of biocatalyst, which comprises of you cultivate the bacteria and you immobilize your bacteria. Secondly, actual hydration takes place and it is subsequently followed by downstream processing, which is catalyzed, is being recovered, and the product is decolorized. Once the product is being formed by this procedure, wherein you first isolate your organisms, okay, make it immobilize, and then add your substrate. That substrate is catalyzed by this enzyme and it forms the product. And after the product formation, what did I tell? It is all your downstream processing. So whatever purification steps is required, whether you're going to centrifuge it, dry it, or decolorize, recovering, all is done by your downstream processing. So another example is the conversion of lignocellulose as biofuel. Okay, so you know now today the cost of the fuels are very high. Okay, it is like skyrocketing. So now what is the another means is okay to produce biofuels by using biological substrate, and for that what they are using is the lignocellulose. So the lignocellulose, that is your plant products, and there are many grasses now grown for the production of only lignocellulose. Okay, a variety of microorganisms involving bacteria and fungi have the ability to degrade this cellulosic biomass to glucose monomers. Otherwise you can go for pre-treatment also. But if there are microorganisms which can directly degrade the lignocellulose into monosaccharides, well and good. You are removing that pre-treatment step. That will lessen your cost also. You can either go for pre-treatment and then the microorganism can act upon it. Otherwise, you be you screen and select an microorganisms which can degrade this lignocellulose and produce the smaller monomers like the glucose. Now, bacteriocellulase exists as discrete multi-enzyme complex called as cellulosomes that consists of multiple subunits. So, this enzyme, okay, the cellulase, it exists as multi-enzyme complexes, and these are called as cellulosomes. So the cellulotic enzyme system from the filamentous fungi, especially Trichoderma resi, contains two exogluconase or cellobihydrolase, at least four endogluconase and one beta glucosidase. Now, what are these all? These are all enzymes. Okay, so the cellulotic enzyme system from the filamentous fungi, that is Trichoderma resi, contains two exogluconase or it contains cellobihydrolase. Okay, this cellobihydrolase, it is CBH1 and CBH2. What are these? These are enzymes. What type of enzymes these are? These are multi enzymes. Okay, and also it has four endogluconase. Okay, so it is mentioned as endogluconase G1, endonuclease EG2, EG3, EG4, and one beta glucosidase. So these enzymes act synergetically, that is together, all these enzymes, no, whether it is your exogluconase or whether it is your cellobihydrolase or your 4 endonucleogluconase, all and one glucosidase. See how many enzymes are involved? One here, two, three, four. Apart from that, they are multi enzymes. Okay, so these enzymes act synergetically. They, that means that act together and catalyze the hydrolysis of the cellulose. So all these enzymes together, they act synergetically together. Okay, In a synchronized manner they act, and they can bring about the hydrolysis of the cellulose. So even different physical parameters such as your pH, temperature, absorption, chemical factors, like you have to provide nutrition to your microorganisms, right? Like nitrogen, phosphorus, phenolic compounds. The microorganisms produce toxic compounds such as the phenolic compounds, and other inhibitors can critically influence the bioconversion of the lignocellulose. So what are we talking here, students? We are talking about the bioconversion. And you spoke here about the bioconversion of this rhamnolipids. Okay. So we saw that it is using this renewable energy to produce a greenwashing detergent, and which is not going to add any wastage to your environment or doesn't cause any greenhouse effect also. Now, next we spoke about the acrylamide. We saw that acrylamide was usually synthesized by the chemical process, but later on we came to know in 1985 there is an organism, Pseudomonas chloranthus, which has an enzyme which can convert a particular substrate into an acrylamide, and it is 50% more acrylamide was produced. And next, and then what they did, we spoke about, was we told that this particular microorganism or that enzyme can be immobilized such that the recovery process can be simpler, and you can reuse this enzyme as many times as you want. Next, what we are studying is conversion of the lignocellulose. Okay, so we told that earlier that lignocellulose are pre-treated by chemical methods. Now there are organisms which can break down the cellulose compound into the simpler monomers. Okay, and here we see that there is a group of, or instead of telling group, let us say there are multi enzymes which are involved, especially like trichoderma. Okay, what they are doing, they are bringing about synergetically to catalyze the hydrolysis of the cellulose. Now all the factors, the physical factors such as your pH, temperature, then whatever nutrition you're supplying, okay, all this influence the bioconversion of the lignocellulose. So the production of cellulose by microbial cell is governed by the genetic and biochemical controls, including induction, catabolite repression, or end product inhibition. For this, you please have to go for this my starting videos of fermentation technology, wherein I have explained in detail what do you mean by this catabolite repression or end product inhibition. Okay. So several factors have been made to increase the production of cellulase through a strain improvement by mutagenesis. So by using genetic engineering, what now the scientists have done is they have improved the strains. Strains means what? The strains of microorganism, so that they can produce more amount of cellulase such that they break the cellulose. Which cellulose? Lignocellulose. Because break the lignocellulose is a very complex compound. It is not very easily degradable. Even only few microorganisms have these cellulase enzymes which can be broken down, which can break down this lignocellulose into smaller compounds like glucose. Therefore, what genetic engineers are doing is they are modifying the strain such that the strain means microorganisms, that microorganism produce more cellulase enzymes so that it can degrade the cellulose into smaller monomers such as the glucose. So celluloid bioconversion is a complex process and requires synergetic action of these three enzymatic compounds what we mentioned: endogluconinase, exogluconase, and beta-glucosidase. The co-cultivation of microbes in fermentation can increase the quantity of the desired compound of cellulase complex. So this is the about the bioconversion. So in bioconversion, what is happening? The microorganisms, that is metabolic process occurring in the microorganisms. They are producing certain enzymes which can convert the substrate into another uh, product or novel product. So what we can do is We can isolate such type of microorganisms, and we can improve them genetically by either by strain improvement or mutagenetically, so that they produce more such type of enzymes, so that these microorganisms, see, they have so much of multi enzymes present, which can go and act on this lignocellulose and break them into smaller monomers, so that these glucose substances which are being produced can be used by other microorganisms for producing any other compounds or the valuable products. And how these are all done? They're all done by now by the fermentation technology. Okay, whenever I say fermentation technology, you have to remember that it is, we I told you the scaling up from small scale to laboratory scale to pilot scale to ferment, what commercial scale or the production scale. So this is the two difference between the biotransformation and bioconversion, which are in most of the papers and textbooks are interrelated, but they are two different terminology, and they have two different functions to perform. So it is very important for you all to write the definition of this transformation and bioconversion, and also you need to write at least two examples of these two methodologies, whether it is biotransformation or bioconversion, how they are bringing about the product formation and how it is different from your chemical process. That should be mentioned. And this is, see, by bioconversion method only they have produced the flavors and fragrance compounds. Okay. These are the precursors, these are the microorganisms, and you can see the different type of flavor compounds which have been produced. This is also an important questions for most of your examination of, like, what are the flavor compounds being produced, and you have to write about the microorganisms which are involved. See, this is the difference I've showed between the biotransformation and bioconversion from one of the papers in Science Direct. So both renewable substrates are used. So this is your microorganism. Okay, it is acting as a biocatalyst here, and the microbial biomass, you get the total biomass here, and it can convert the heterologous enzymes into product. That is, whatever preformed substrate, whatever the substrate was there, I told you, no, steroid is there, and it is getting converted into the other form of testosterone, or I told you what is that, cholesterol. Okay, that is like this. In bioconversion, again, you have renewable substrate, microorganisms, which produces your biomass, and then here you can see it produces the intermediates, which through the help of the enzymes, it gets converted into the product. There is nothing called the preformed substrate here. So whatever the biochemical nutrients, as I told you, the first synthesis of biochemicals from the basic nutrients from the microorganisms. So whatever the reactions is going on in this microorganisms, it consumes the nutrients, that is known as the cellular metabolism. This is cellular metabolism happens in that body because of the enzymes, and we can make use of these enzymes. And also there are quite a lot of application of this biotransformation. See, it causes conversion of active compounds to less active compound, which is called as inactivation or detoxification. For example, DDT to metabolite product DDE and DDA. You know the DDT is very harmful, carcinogenic, but what it is doing, it is converting into less toxic form. Okay, and therefore It is called as inactivation or detoxification. In the second, you can see that it causes conversion of active components to more active metabolites, which is called as bioactivation. This is inactivation, this is bioactivation. Example are malathion to acetonitrile to cyanide. It causes conversion of inactive compounds, that is prodrug or precursor, to active metabolites, which are called as activation. Example, phenacetin to paracetamol and thiocyanides to cyanide. So it is bringing about the conversion of inactive compounds to the active compounds. Fourth, it causes conversion of active compounds to an equally active metabolite. Example, digitoxin to digoxin. So here what happens is equally active metabolite without no change in the activity, you are getting the another good compound. Just convert this compound to another compound. It causes the conversion of active compound to active metabolites with entirely pharmacological and toxicological activity. Example, aflatoxin B1 to aflatoxin M1. This is hepatotoxin and this is a carcinogen. So these are some of the functions of your biotransformations. Okay, so you can see that the toxic compounds can be converted into non-toxic forms. Otherwise, the inactive metabolites can be converted into active metabolites. Or you can see that it causes conversion of active compounds to more active compounds here. And here you saw the conversion of the active compound to an equally active metabolite without any change in the activity. And here we have seen the causes of conversion of active compound to active metabolites which are having pharmacological and toxicological activity. So students, in this section you studied about the biotransformation and bioconversion, and also you saw the difference between these two terminologies. And finally, we have ended up telling what are the functions of these biotransformations. And for each, we have given two or more examples. So with this, we come to the end of the uh, this section on biotransformation and bioconversion. So both are equally important when you compare it to your chemical procedures. Okay, you can see how environmentally friendly they are, and also the cost of operations can also come down by using these two methodology. In both these methodologies, what we are concerned was about the microorganism, how these microorganisms are converting these product into more useful product. If already a substrate is there, how I can convert it into a more novel variety. Okay, otherwise we saw that how by cellular metabolism, what the nutrients the microorganisms are consuming, the metabolism which are happening, it happens through enzymatic reactions. And through this enzymatic reaction, how this biochemical synthesis of certain compounds can happen. With this, we come to the conclusion of this chapter on the bioconversion and biotransformation. You have to especially remember the